So we also categorize our samples depending upon which day we receive them. So if you send a sample on a Monday and we receive it Tuesday, that will go there in Tuesday, so that if we have to go back, we are not looking for the sample. So we are kind of like very systematic here. And some days you get more samples, some day you get less samples. But we generally recommend that if you are interested in submitting a plant sample, you want to do it early in the week, so we have time during the rest of the week to diagnose that problem. Your sample doesn't have to sit over the weekend here at Diagnostic Center in order to be processed. So once we once we plate the affected leaf tissue or the root tissue on the artificial media, then we bring them over here and we store them at a specific temperature so that the pathogen can start growing out of it. Here is a good example from hemp, industrial hemp. The plant was infected with southern blight, and once we grow it on it, and this is kind of a classical example where the southern blight is producing the sclerotia, okay, a mustard seed size and shape and color sclerotia in the plate. Once we see this, we are about 80% sure that this is southern blight. Okay. Again, we have to make sure that we have what species. There are different species of the same genus, so we have to figure out based on molecular test like sequencing, DNA sequencing, on what that particular species is. So this is my staff here, Mr. Tim Burks and Dr. Monique D'Souza. Dr. D'Souza started working with me last year, and I'm very fortunate to have her. And Mr. Tim Burks over here, he has been working with me for how long now?
2012.
And he is our lab manager, and he takes care of most of your samples here. Okay, I just send you the report, so he's the main man behind all these scenes. Okay.
I'm gonna.
It's okay. So this is where we culture our pathogens. So you can see here some plates that have produced the mycelium that we're looking for, or the fungal pathogen. Yeah, this is actually not a fungal pathogen. It's fungal-like microorganism, which is Phytophthora. Okay, it's basically known as a water mold. Here in this room, we do the gel documentation system. So once we do the DNA extraction, we amplify the DNA, but you can't see the DNA with naked eye. But here in this room, what we do is we run those DNA samples, and you can actually see the DNA. So we use this is called gel documentation system. So once we put all the cameras on and everything, and you can actually then see. So this is a new pathogen that we are dealing with here. The name of the host is American hornbeam. It's a very native tree here, very popular tree here in the wild. And here you're looking at two samples of DNA, and this is the DNA amplified. Now you can't see that with naked eye, so you have to use this equipment in order to see the DNA from that plant material. And you can see this is a really nice gel picture that is showing you two samples, two duplicates, and then two duplicates. So this is a very good DNA amplification here. This is our PCR room where we run the conventional PCR and the real-time PCR, and this is our real-time PCR machine here. The difference between these two PCRs is that the real-time PCR is in real time, so you can see the results while this machine is amplifying the DNA. And the conventional is that you have to amplify the DNA first, and then you have to run the gel documentation system on it and see the results on the gel. In this case, you don't need a gel to run after you run the PCR. So this is an example of one of the citrus canker sample that we have here. So you see here three lines. Okay, so what you are seeing here is two samples. One is our positive, which is this blue line, Okay, and the other one is our sample, and you can see it amplified after 21 cycles. Okay, that is a positive, that is considered a positive sample. And here you can see the results. You have the water, which is our negative control, the positive, which is on the another control, and down here so for the sample to be a positive sample, it has to be in a range. It can't be after 35 cycles. So if it's after 35 cycles, that's not considered a positive sample. So we are still running a conventional PCR here. In this case, there are 3 different steps that it will run for several, for I would say 40 steps, and each cycle has 3 steps in it. So you can see it goes to 93 degrees Celsius, and then it comes down to 64 and then 72. So this is called denaturation at 93, and at 64 it's annealing those building blocks. So it's making the DNA, amplifying the DNA, and then this is at the last step is 72. Okay, so and then it will repeat. Okay, it will repeat, it will go back, repeat, and you can say it's going to repeat here on this particular test that we're running. We are running this for HLB, which is your citrus greening disease. So this will run for 34 cycles. It will run for 34 cycles with these 3 steps, and then we will go back, use the gel documentation system to see if it amplified the DNA or not. These 2 workstations are our clean workstations. We use them to mix our reagents. One is for making the master mix, and the second one is adding the DNA template that we isolated from the plants, from the leaves of the plants, plant tissue.